which is cell growth and cell division. Now up here has a picture of a cell that is in the process of dividing into two cells. This is probably an animal cell, and the reason I can tell that is because it does not have a cell wall. Alright, anyway, now let's talk about cell division a little bit. In eukaryotic cells, genetic information is passed on from one generation to another by chromosomes. Now, chromosomes made up of DNA, remember deoxyribose nucleic acid, and we have 46 chromosomes inside our bodies. Now, what would be the purpose of mitosis? The purpose of mitosis would be to reproduction in unicellular organisms. It's a type of asexual reproduction. These are both actually saying the same thing. But for us, the big reason why we do mitosis is right here: growth and repair. So when I think of mitosis, I think same. If I were to cut my arm and those cells would have to, my arm would have to close back together where it got cut, it would grow, the cells would grow by a process of mitosis. Mitosis makes the exact copy of the cell that's there. So now if we look at asexual reproduction, if I were to reproduce by mitosis, there would be another Mr. Kennedy sitting beside me. It would make an exact copy of me. I know that's a scary thought. Alright, now I know a lot of you haven't actually seen a chromosome before. This is a picture of a chromosome. There is something fictitious here. They've superimposed a DNA molecule into this picture. But this is just simply a chromosome, kind of been stained with something like bromethyl blue like we use with our plant cells when we looked at them under a microscope. Anyway, now don't get confused about mitosis and the cell cycle. They're not exactly the same because mitosis is actually a part of the cell cycle. The cell cycle is a series of events that we go through: growth and division. Now, the cell cycle, basically, here's what happens. The cell grows, prepares to divide, and it divides into two daughter cells. Then those two daughter cells grow, prepare to divide, and divide into two daughter cells. And then those daughter cells grow, prepare to divide, and divide. So it's just a continuous process. Now, this first step, I'm going to list for you the steps of the cell cycle. There are 5 steps of the cell cycle. There are 4 steps of mitosis. Mitosis makes up the cell cycle with the addition of this stage, interphase. Interphase has 3 different phases: the G1, the S, and the G2. We're not going to really be concerned about those in regular biology. If you ever take AP biology, we'll get into those. But here's what you need to know for the test. Interphase is where the cell grows, it gets bigger, and chromosomes replicate. That means if we have 46 chromosomes in our cell, once we go through interphase, our cell is bigger and it has 92. Now let's think about why this is. Mitosis is going to make when we go through mitosis, we're going to make an exact copy, and I told you the last 4 steps of the cell cycle are mitosis. So if we're going to make an exact copy of the cell, if we start out with a cell and we won't make an exact copy, we've got to copy everything in that cell, the DNA, because when we split it, we want half of it to go one way and half of it to go another way, and that's what's going to occur. Now let's look at the 4 stages of mitosis. Remember, this is stage 1 of mitosis, but stage 2 of the cell cycle. The first one is prophase. Prophase is the longest phase of mitosis. Interphase is actually the longest phase of the cell cycle. Here's the things you need to remember. Spindle fibers form. You don't really know what those are yet. They're right. You start seeing them form over here. You don't know what they are yet. The nuclear envelope disappears. That's the membrane around the nucleus. And the chromosomes coil up. Now I like thinking like this. The cell is preparing for its chromosomes to separate to opposite ends of the cell, or to go on a trip. If you were getting ready to go on a trip, first thing that would happen is you would pack your luggage, because by packing and folding your clothes neatly, you're able to pack more clothes inside your luggage bag. So that's what the chromosomes coil up is. It's packing the chromosomes. Next thing, if you're going to separate the chromosomes and DNA can only be found in the nucleus, then you have to eliminate the nuclear envelope so that the DNA can be separated. So the nuclear envelope disappears. The last thing is spindle fibers, and I'll tell you what that means on the next slide. Metaphase, chromosomes line up across the middle of the cell. Now, notice right here, you see these lines? These lines coming out from each end are the spindle fibers. Now I kind of like thinking of it like this. And by the way, these right here, that's the chromosomes. I kind of like thinking of it like this. If I had a fishing rod that had a treble hook at the end and I were to throw it out and hook you behind the ear with it, if I started reeling, you would come to me. That's kind of what the spindle fibers are doing. They throw out their treble hook and they hook onto a chromosome, and they're going to pull some chromosomes this way and some chromosomes this way. Now think about it. If they copied, they're going to pull a number 1 this way and a number 1 this way, a number 2 this way, a number 2 this way, a number 21 this way, a number 21 this way, a number 23 this way, a number 23 this way. You know, so you end up with exact same thing on both sides of the cell. The next phase is anaphase, and as you can see in the picture, the chromosomes have started to separate. Here's the chromosomes, right? Here's the chromosomes. These chromosomes are going this way, this chromosome is going that way because these spindle fibers here and these spindle fibers are pulling them. So sister chromatids separate. Sister chromatids just means that the copies you made, one copy goes one way, one copy goes the other way. Then telophase occurs. Notice what happens. Chromosomes uncoil, the nuclear envelope disappears, and spindle fibers disappear. So, nuclear envelope reappears, I'm sorry. Spindle fibers disappear. This is the opposite of prophase. Why? Because they've reached their destination. The chromosomes unpack, the nucleus reappears, or the nuclear envelope reappears around those chromosomes, and the spindle fibers disappear. That's telophase. All right, and the last phase is cytokinesis. Now, sometimes you'll see this linked in with telophase. Cytokinesis, cyto means cytoplasm, kinesis means to split. So cytokinesis is splitting of the two cells. All right, now let me show you this in just a minute. If I was to say the cell cycle would be IPMAT is how I remember it. Interphase, prophase, metaphase, anaphase, telophase. If I was to say mitosis, it would simply be PMAT. It's missing this I. So remember what happens. Chromosomes duplicate during interphase and the cell grows. Prophase, what happens? Prophase, 3 things happen. The chromosomes coil up, spindles form, and the nuclear envelope disappears. Metaphase, I think middle. The chromosomes line up down the middle of the cell so they can be separated. Anaphase, chromosomes go to opposite ends. All right? And in telophase, we get 2 cells being created by a process of cytokinesis. And the opposite of prophase occurs. Chromosomes unpack, uncoil, nuclear envelope reappears, and the spindle fibers disappear. You need to know what happens in each stage. You need to know these stages in order for the test and be able to identify pictures that may be associated with these phases. All right, I hope this helps you to understand the cell cycle a little bit better and the difference between cell cycle and mitosis. And have a good day.